Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's dive right in because this is wild. Forget what you thought you knew about bacteria spreading resistance genes. Scientists just found this shocking new kind of microbial piracy.
Yeah, it really turns some things on their head.
Imagine this tiny viral parasite, right? It
Yeah.
builds its own head, the capsule with its DNA, and then just releases it, tailless.
Mm-hmm.
And here's the kicker.
Yeah.
It then steals a tail from a completely different unrelated virus to infect a whole new type of bacteria.
Exactly. This modular hijacking by these elements called cf-PICIs, it's basically rewriting the rules for how things like antibiotic resistance genes can jump between bacteria.
So that's the core claim right there. We're digging into a cell paper and some analysis on it today, our mission. To unpack this tail piracy, figure out how it works, and why it's such a game changer for tracking antimicrobial resistance, or AMR.
Precisely. This research plugs a really significant hole in our understanding of how these mobile genetic bits get around. It's this incredibly clever evolutionary setup.
Okay, so let's set the scene a bit first. The background is horizontal gene transfer, HGT. Basically, bacteria swapping important genes, right? Like for resistance or making you sick.
Yep, that's the microbial gene swapping network. And often, bacteriophages viruses that infect bacteria are the delivery guys.
Got it. So phages are the usual suspects for moving genes around.
Right. And within that world, you have these things called phage-inducible chromosomal islands, or PICIs. Think of them as classic parasites living on the bacteria's chromosome.
Okay.
They normally hijack a helper phage, completely make it package and spread their DNA instead of the phage's own.
Standard parasite stuff.
Kind of. But this paper focuses on a subgroup, a really common one, the cf-PICIs. Capsid-forming PICIs.
Capsid forming, meaning they build their own heads.
Exactly. They make their own DNA containers, their heads are capsids, but crucially, they're born without tails.
Huh. So you've got these super common genetic elements found everywhere, but they're missing the part they need to actually, move and infect. That sounds counterintuitive.
It was a huge puzzle. These cf-PICIs are wildly successful, found in over 200 bacterial species, which implies they move around a lot.
But how? If they don't have tails?
Well, the standard thinking assumes a really tight, specific match between a phage and its host for transfer. But when the researchers first tested this with the known helper phage, P1, the results were terrible.
Terrible how?
Almost zero transfer, even within the same bacterial species, and absolutely no transfer between species, even though the cf-PICI DNA was getting packaged into heads.
So the heads were being made, the DNA was inside, but they just weren't going anywhere effectively.
Correct. That failure was actually the key. It proved the resident helper phage, P1, wasn't the answer for crossing species boundaries. Something crucial was missing.
So if the helper phage wasn't providing the right legs, what was the hypothesis? How are they getting around?
They figured the problem might be structural. The helper phage they used, P1, is a potovirus. It has short tails. But the cf-PICI seemed to need long tails, like those from several viruses, to work properly. So they took a closer look with electron microscopy.
And what did they see?
Two really important things. First, the particles being made were definitely tailless capsids, just the heads, full of cf-PICI DNA, no tails inside.
Confirmed tailless. Okay, what was the second thing?
Their size. The cf-PICI capsids from this kPCI1 element were small, about 45 nanometers across.
And the helper phage? P1.
Much bigger, around 65 nanometers.
Ah, wait. That size difference is key, isn't it? Does being smaller, that 45 millimeters, stop the cf-PICI from accidentally packaging the helper phage's much larger DNA? Like a safety lock.
Exactly. It's a built-in physical defense. The smaller capsid, it has what's called T4 symmetry, and it's just too small to fit the helper phage's genome, which needs a larger T7 shell.
Clever. So it ensures only its own cargo gets loaded.
Precisely. So the big takeaway here isn't failure, it's strategy. The cf-PICI is intentionally making and releasing these non-infective specialized tailless heads, ready for something else.
Right. This is the aha moment then. if they're releasing tailless heads, the obvious, well, maybe not obvious, but the logical next step is they must be finding tails somewhere else.
That was the theory. Docking onto free-floating, compatible tails in the environment. Tails left over from totally different phages. It's like biological scavenging. Or piracy.
Okay, so how did they prove that? That sounds hard to show.
They set up a critical experiment. They took those purified, tailless KPCI1 capsids, remember these originated in Klebsiella pneumonia,
Got it, a specific bug.
and they mixed them with just the tails stripped off an E. coli phage called HK022. Completely different source for the tails.
So heads from Klebsiella, tails from an E. coli phage, mixed together.
And boom, they created a chimeric infective particle. It worked incredibly well.
Oh, well.
They got super efficient transfer of the KPCI1 into E. coli bacteria. We're talking a frequency of about 2.3 times 10 to the 7th colony-forming units per milliliter. That's huge.
Whoa, okay, that's not just a little bit of transfer. That's highly effective transfer between different genera Klebsiella to E. coli. That's revolutionary.
Absolutely staggering efficiency. The borrowed HK022 tail essentially acted like a key, dictating the new host E. coli and letting the KPCI1 capsid sneak its DNA inside.
So the head carries the cargo, but the tail determines the address.
Exactly. The cf-PICI basically gets to use the host range of whatever tail provider it finds. Doesn't matter where the head originally came from.
It's the ultimate plug-and-play host switch, like you said. Or maybe like renting a specialized delivery truck just for the last mile. It doesn't build its own truck, it just flags one down.
That's a great analogy. And they also confirmed how it gets the tails. It's not ripping them off fully formed mature phages.
Oh, how does it get them then?
It grabs unassembled tails. During the replication cycle, phages often make way more tails and heads than they actually need. There's this big pool of spare parts lying around in the environment.
Leftovers, basically.
Exactly. The cf-PICI is just smartly using the abundant spare parts. It's not fighting mature viruses for their tails.
Yeah.
Much more efficient.
Okay, proves it in the lab. Mixed heads and tails get infection across species. But the big question then is, does this actually happen out there, in the real world, in a messy microbial soup?
All the signs point to yes. They found these tailless capsids are produced naturally, spontaneously, even without a classic helper phage kicking things off. Just by stressing the bacteria. Things common in nature like antibiotics or lysozyme, something that damages bacterial cell walls. Stress the cell, it releases these tailless heads.
Wow, so environmental hardship actually triggers the release of these potential gene bombs.
Seems like it. And they demonstrated this ecologically too. They set up a co-culture, like a little ecosystem in a tube.
What was in it?
3 different bacterial strains. One donating the cf-PICI, the head maker, one providing the right kind of tails, and a third one as the recipient target. All from different species growing together.
And did it work? Did the cf-PICI transfer?
Yes, they saw efficient transfer happen under those more natural conditions.
Okay, that's compelling lab evidence for natural occurrence. What about, like, really in the wild?
Even better, they looked at real-world data from human metagenome studies specifically. Virome samples, the viral parts of our microbiome.
Yeah.
They found packaged cf-PICI DNA in there, in samples from places like the human gut. This strongly suggests this tail piracy mechanism is actively happening inside us right now, shuffling genes around.
Mind-blowing. It's happening in our gut microbiota. Okay, but let's talk specificity. If it's just grabbing spare tails, is it totally random which tails it grabs and which bacteria it can then infect? Or is there some control?
Definitely not random. It's actually quite specific, and the control lies within the cf-PICI itself.
How so?
The cf-PICI encodes its own special proteins called adapter and connector proteins. Think of these as the interface, the docking mechanism on the head.
Ah, so the head has proteins that determine which tails can successfully attach, like a lock and key.
Exactly. These proteins dictate which specific phage tails the capsid can hijack. For example, the KPCI1 element they studied could only use tails from the HK0202 phage.
Okay, specific to that one tail type.
But another one they looked at, ECI1 from E. coli, was more flexible. It could use tails from both HK022 and another phage, HK106.
Fascinating. So different cf-PICIs have evolved different levels of specificity for the tails they can pirate. It's targeted modularity.
Precisely. Highly evolved, targeted modularity. The cf-PICI isn't just grabbing any old tail. It's evolved the specific connectors needed to grab the right tails for the job.
Which brings us to the structure.
Yeah.
How is the head itself built to enable this whole pirate strategy? You mentioned cryo-EM.
Yes, they solved the structure of another cf-PICI capsid, ECI2, at really high resolution. And it reveals how it defends itself. It sets itself up for piracy.
What did they find structurally?
We already mentioned the size difference, a T4 symmetry making it small, only 47.5 nanometers, much smaller than its helper phage, HK97, which is Tcl7 and about 60 nanometers. That size difference physically prevents packaging the larger phage genome.
Right, the built-in safety lock. What else?
They looked at the major capsid protein, the main building block of the shell. ACI2's protein has this unique connector loop region. They think this loop might actually help force the structure into that smaller T4 shape instead of the default T7 that related phages use.
So a specific protein feature helps ensure the small self-packaging size.
Seems likely. And another key finding was about stability. It's relative, the phage HK97 uses strong covalent cross-links like permanent chemical bonds to make its capsid really tough.
Okay, like reinforcing steel?
Right. But these cf-PICI capsid, CCI2, it's held together entirely by non-covalent interactions, things like salt bridges and hydrogen bonds, weaker, more temporary bonds.
Why would it do that? Seems less stable.
Possibly because it needs more flexibility. Maybe it makes assembly easier, or perhaps it relates to how it docks with the tail later. It's structurally distinct, optimized for its specific parasitic modular lifestyle, even if it seems less robust on paper.
So putting it all together, this whole mechanism is huge. It just shatters the old idea that a mobile element like this is tied to just one specific helper phage for its mobility.
Completely. Tail piracy makes gene transfer modular. It's scalable, just find more tails. And it can probably bypass a lot of barriers that normally stop phages, like thick bacterial capsules or biofilms, because the tail provides that final specialized entry mechanism.
Which leads straight to the practical side.
Yeah.
How can we use this knowledge? You mentioned AMR monitoring therapeutics.
Yeah, the translational potential is really exciting. For diagnostics, imagine using this. We know specific tails target specific bacteria, so maybe we could engineer tailless reporter capsids. We release them, they grab naturally occurring tails specific to, say, a dangerous pathogen in a sample, and then deliver a signal like making the pathogen glow only when they find their target.
Using the environment's own delivery system to tag the bad guys? Clever.
And then there's therapeutics, the idea of anti-AMR satellites.
Okay, explain that.
You engineer a cf-PICI. Instead of carrying its own genes, you load its capsid with a payload, maybe something lethal to the bacteria, or maybe a gene that resensitizes a resistant bacterium to an old antibiotic.
So you make a weaponized cf-PICI head.
Exactly. Then you rely on the tails naturally present in the infection site tails associated with phages that already infect that specific resistant pathogen to deliver your payload.
Wow. So if you have, say, MRSA infection, and there are phages around whose tails recognize MRSA, you release your engineered anti-MRSA heads, they grab those MRSA targeting tails, and deliver the payload right into the MRSA cells.
That's the concept. Using the natural phage tail landscape as your targeting system for engineered therapies, it leverages the environment's resources for precision delivery, a potential new weapon against resistance.
It's incredible. We started with what looked like a defective genetic element, and it turns out to be this super sophisticated molecular pirate mastering resource efficiency and gene spreading across huge bacterial divides. It's a biological hack of stunning elegance.
It really is.
Yeah.
And it leaves us with a final kind of provocative thought to chew on.
Oh.
If cf-PICIs show that separate genetic entities, a head from one place, a tail from another, can team up to make a functional infectious particle, could phages themselves do this?
You mean if a bacterium defends itself by blocking its own phage from making tails?
Right. Could that now tailless phage head then get hijacked by other free tails in the environment? Could it form new, unexpected chimeric phages we haven't even considered?
Huh. So even viruses might be engaging in this kind of modular mixing and matching, maybe accidentally.
It opens the door to that possibility. This research highlights that modularity, cooperation, and piracy are powerful evolutionary drivers in the microbial world, maybe creating diversity in ways we're only just starting to appreciate. Our picture of the virus sphere might be far from complete.